Welcome to Dr. Balga's podcast for the Curious Doc. Today is February 18, 2026. This video is on a Nature article published today titled Highly Dynamic Dural Sinuses Support Meningeal Immunity. The dural hosts venous sinuses, traditionally viewed as passive drains. This nature study demonstrates that dural sinuses are dynamic neuroimmune interfaces, regulating intracranial pressure, fluid exchange, and immune surveillance. They used a murine model, a mouse model, and did intravital 2-photon microscopy through thin skull windows, confocal imaging, light sheet microscopy, and microcomputer tomography. They did single-cell RNA-seq of sinus endothelial cells, electron microscopy to identify fenestrations and openings, and for pharmacologic modulation they used CGRP agonism, RAMP1 antagonism, and RAMP2 antagonism. Viral challenge with lymphocytic choriomeningitis using the virus Armstrong. Statistical analysis include ANOVA, mixed effects modeling, and linear modeling. What they find: superior sagittal sinus in the mice is bifurcated into upper and lower chambers, contributing to intracranial pressure regulation. Compression of the upper sinus significantly increased intracranial pressure, p-value 0.0001. CGRP-induced vasodilatation with increased perisinus flow, p-value 0.0001. BIBN 4096 caused vasoconstriction with altered luminal velocity. RAMP2 antagonism reduced sinus endothelial cell ruffling, p-value 0.0337. During viral infection, the ruffles increased, p-value less than 0.0001. And the blockade enhanced viral invasion was associated with a p-value equivalent to 0.0186. These ruffles are transient amorphous openings that form between V cadherin positive sinus endothelial cells along the superior sagittal sinus wall. They represent dynamic remodeling of endothelial borders rather than fixed structural pores. Limitations of the study: primary murine model, it's a mouse model. Human validation limited to imaging and histology. Pharmacologic modulation rather than genetic deletion models were used. The key takeaways are: dural sinuses are contractile, fenestrated, immune active structures. RAMP1 regulates smooth muscle tone, RAMP2 regulates endothelial boundary dynamics and antiviral defense. Clinical pearls: dural sinus compression can rapidly elevate intracranial pressure. Venous sinus pathology may impair immune surveillance. Calcitonin gene-related peptide CGRP signaling influences venous tone. Adrenomedullin receptor signaling modulates endothelial permeability. Venous sinuses may represent therapeutic targets in intracranial hypertension, infection, and neuroinflammation. There is an accompanying news article in Nature titled How Skull Drains Keep the Brain Safe from Damage and Pathogens. It discusses the key discoveries: that the sinuses are wrapped in smooth muscles, constrict and dilate to pump blood and cerebrospinal fluid. Endothelial walls contain fenestrations about 1 micrometer, allowing passage of fluid and macromolecules. The unique behavior called ruffling. Endothelial borders rearranged to accommodate patrolling immune cells. Blocking RAMP2 impaired antiviral defense, making sinuses behave more like passive vessels. CGRP dilated sinuses, suggesting migraine therapies may partly act via sinus modulation. Why it matters: the brain's borders are not static barriers but regulated, adaptive interfaces. Dual sinuses actively protect against pressure changes, inflammation, and pathogens, functioning less like plumbing and more like vigilant gatekeepers. Read this fascinating news article in Nature published today. This study is about the RAM proteins. Hit pause and read at your own leisure. And this slide is about um, eponyms related and concepts related to this study. Hit pause and read at your leisure. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.